Okay, I'm going to be presenting not really a data talk, but much more of a synthesis from sort of my own experiences and experiences of people I know working for about the last 10 years, trying to understand from resurvey type studies how species shift their range and how we can detect this. You're welcome to tweet this. My Twitter is my first two initials and then the last name, M.W. Tingley. So the foundations of inference in science is based on taking samples, multiple samples. And in a generalized sort of Popperian framework, we make inference because we use these replicates to generalize about a repeated condition. So we assume that both of these samples, for instance, are taken from randomly from a true population, and then by looking at the conditions of these two samples, we can understand that true population. With climate change research, what we're doing is we're using replicates to describe what happens with a changing process. And Greta talked earlier, kind of now famously over Twitter, by describing climate change as a poorly planned experiment. And I would like to take that one step further and say it's not an experiment at all. We do not have controls. And so without controls, we can't call it an experiment, and it means that inference about climate change impacts is very, very difficult. And so imagine we wanted to understand the change in font size of two samples over time. And so in this typographical example, we might say that we can imagine the change in the font size, but the problem is that other things have changed at the same time. So we've now moved from nice square corners to rounded corners, and actually the font has changed from a sans serif to a serif, which means that understanding these processes of font size change over time is very difficult because other things now get in the way. And that's discounting the fact that when we think about change over time from climate change, it's not the only thing that's happening in our landscapes. In terrestrial realms, we have afforestation, deforestation, grazing, changing in fire regimes. If you work in the marine world, you have overfishing everywhere. You have trophic cascades, pollution, restoration, non-equilibrium dynamics, and endocycles. And all of these things compound to make the fact that our inference of change over time from climate change is quite difficult. So sort of the two major questions that I see in terms of how we analyze data to understand climate change impacts can come down to two synthetic topics. One is how do we control for bias in our comparisons that arise from changing sampling conditions? And the second is how do we properly attribute observed changes? I don't have time to talk about both of these, so I'm going to focus just on this first one, about how we can control from biases in these comparisons over time. And so to give an example of when things can go problematic, I'm going to return to a paper that Camille talked about early this morning, which is John Crimmins' 2011 science paper, which looked in California, looking at changes in range shift dynamics over time using herbarium specimens. And this showed sort of overwhelmingly species were shifting downslope throughout California and the plant world. Fast forward 4 years now, it actually came out this week, a paper by Adam Wolf and others used essentially the same data set and showed that overwhelmingly in California, plant species had shifted upslope. They relate this total change in reversal and trend to a paper they published 2 years ago, which looked at drastic changes in sampling effort over time in California and vegetation, and that this change in sampling effort can give rise to these differential conclusions about whether or not plants were shifting upslope or downslope. And so I'm not going to take sides on which one is right, but to point out the fact that we need to be doing this right. And to the extent that we can do this right and try and make inference when our sampling is often poor will sort of help us out when we're trying to communicate our science to the broader world. So I've distilled this down to 5 steps for better comparisons that I'm going to go rather quickly and then talk about each one in turn. The first is know your data. The second is match conditions as much as possible. The third is compare using robust metrics. Account for differences statistically, don't hide them, and repeat. The more data, the better. And if you're writing this down, I'll come back to this at the end so you get a chance to finish writing them. So I'm going to draw some examples from my own experience working in the Grinnell Resurvey project in California, resampling at specific sites in 3 elevational transects across the Sierra Nevada in California, in my case focusing on birds. And so my first point is know your data, and this might seem simple and obvious, but the point is that when we're trying to make comparisons over time, we have to account for the biases. And when we're making these long-term comparisons, the point is that often we were not the people that went and collected the data in the first place. And since we did not collect the data, we do not know the data intrinsically. And if we do not know the data, then we cannot compare, we cannot account for all the problems in how those data were collected. So we have to go back, and we cannot just use online databases that have been handed to us to understand understand how species are changing the range. We have to go back and look at the conditions as much as we can, find the field notes, find the records, find the metadata about how these data were collected in order for us to go back and make sure we can fairly account for biases. The second is match conditions. Now, we want to try and, obviously things are collected in different ways over time, and we have to try and account for those changes. Now, we need to know what's important to match and what's not. So if I'm going to be going out and resurveying birds from 100 years ago, I'm not going to deliberately handicap myself by using antique binoculars. But some things are important to match, and so space and location is certainly one of them. This is a sort of a cute example we had where I worked with an undergrad to survey birds on the Berkeley campus. And we went back through the historical records and accounts and journal entries to find the exact route through the Berkeley campus that historical bird survey people had done, because we wanted to make sure that we were surveying the exact same parts of the campus over time, so we didn't have some sort of spatial mismatch in our comparisons. Another thing that's important to keep track of over time is time. So when you're doing surveys. In this case, with our work from the elevational gradients, both historically and clear and modern and our gray, they were sampled in different times of year, and we wanted to make sure that actually within elevational bins that we were sampling the same sites around the same time of year, because phenology happens differently with elevation, and we wanted to make sure that we weren't missing birds or getting extra birds because of phenological mismatches in terms of arrival times of migratory birds. So things like space and time are very important to match when we go back and revisit sites and make comparisons. The next thing we want to do is we want to try and compare using a robust metric. And so there's this question of which data do we use and what do we want to do with it. So when we have historical data, there's often a lot of data, and we have to make choices about which of these data are good and which of them are bad. So if you look at this historical data sheet, this is from Tracy Storr surveying Yosemite National Park in 1916, you can see some things that might be warning signs, mostly question marks. So were they really 2? Was it just 1, or was it 1+? Was this actually a Sierra crossbill, and was this a right flycatcher or not? So things like question marks and scribbles and X's are usually warning signs that maybe we should not be using the data. And so when we're trying to filter out the data a priori, not after we analyze, but before we do any analysis, trying to figure out, well, which of these sites are actually extreme sites, and maybe they were only visited a few times, or they were visited ad hoc, which survey events were very late in the season, very early in the season, or was the weather really terrible on that day? Particular scientists, often multiple people are collecting data, and some of them actually, honestly, are not very good at collecting data. And you might figure that out from looking at their data sheets. Particular species, really rare species, or species that you might not have had good survey methodology for, you might want to cut out ahead of time. And then certain collection methods as well. If these data were collected at the same time as shotgun surveys, then you might not want to collect use the shotgun surveys in the same way that you use sort of systematic bird surveys. So we're going to be filtering data to sort of combine them and sort of use only the strongest data we have. And this often focuses on trying to find the extremes in your data set in the same way that you would remove outliers. We should be removing essentially outlier historical data before we make any sort of comparisons. And then another thing we can do is standardize. And so we can standardize and often sort of reduce data quality, reduce data density, I should say. So including things like, so I like to do changing abundance down to occurrence data. So often we collect data as abundance data. That doesn't mean we often collect abundance data very well. Vast quantities of the wildlife world are based on the fact that we do not collect abundance data well. And so, looking at the historical data, we have lots of question marks about just how many they were, and probably these abundance data were not collected in a very systematic way, very carefully matching exact amounts of time or exact amounts of space. And so these abundance data we probably should not be using. They're not systematic enough for us to be making comparisons, so we should reduce the data density and reduce them down to occurrence data, because that's probably going to be a more robust metric to the observational biases. The next thing is to model differences and incorporate those differences as much as possible. And so I'll just give a shout out to this paper we published in 2009 in Tree that's sort of laying out a framework for how you can do this in a systematic way. And so what I like to do is take a state-space approach and assume the fact that there is a true underlying state, whether it's abundance or occurrence or something else, that we're trying to understand, and the fact is that we are imperfectly observing it, because historical data are very, very much imperfectly observed. And so we can imagine having sort of going to the same spot in 2 points in time, and you have 2 different bird species, because I study birds, and you visit in each point in time, you visit them 3 different times, and you look at what is the occurrence patterns or the detection pattern across those 3 visits. And so we can imagine with this ruby crown kinglet, you visit the site 3 times and you find it all 3 times. You don't find northern goshawk. You visit this site 100 years later, you don't find any ruby crown kinglets, and you find northern goshawk 1 time out of 3. And so taking a state-space approach for this type of data uses something like occupancy modeling. So occupancy modeling, I know they talked about in some of the pre-workshops. It is a very powerful method where we separate out the detection process from the occurrence process, because the occurrence process is what we care about, but we imperfectly detect it. So we use the fact that we have repeat visits or samples over time to try and account for this probability of detection. And we use this modeled probability of detection to model a true underlying probability of occupancy. The power comes from the fact that we can relate different covariates to occupancy and detection, and therefore sort of model different parameters and model changing detectability or occurrence over time. To go back to this example, we sort of a toy example of how this works. Since we found the ruby crown kinglet 3 times out of 3 when it was present, we can then say that we perfectly detect it, probability of detection equals 1, and therefore we can assume that in this other site, when we, in time 2, that the probability it actually occurs here is 0. Alternatively, with the northern goshawk, since we found it 1 times out of 3, the probability of detection is 1/3, which means there's a 30% chance that actually it occurred here historically and we just didn't find it. The actual math for this is a big binomial likelihood, but it's relatively easy to fit these parameters. To take an example of what this looks like from some of our data, we can take 2 different species, and so we have what look like apparent range shifts. So an upward range shift here, an upward range shift here, and a lower range shift here. And so we can go back to these and say, okay, accounting for the fact that we do have false zeros in our data set, what is the probability that this apparent range shift is actually a false range shift? And so in this case, we can be highly confident that this is a true range shift. In the case of the evening grosbeak here, we find out that the upper range shift is true and the lower range shift is probably just a false. What this means is actually what we're doing is we're minimizing, essentially making false conclusions about range shifts. And so this is from appendix that probably no one has ever seen, but it shows through simulations that we are highly minimizing type 1 error rates. So we are not going to say that there is a range shift when in fact there was no range shift. So we are super minimizing essentially across detectability. We had less than 1% probability of making a type 1 error. What we're doing is, unfortunately, that comes at the expense of something, that it comes at the expense of type 2 errors. So we're probably missing range shifts, which I would say is okay. I mean, there's got to be species that have realistically shifted their elevational range by 1 meter, and we're just not going to find those out. And we have to be okay with that, and the stronger thing, the better thing, is to be confident that as we're communicating our science, that we are talking about range shifts that we're confident in. So my final, then, message is repeat, repeat, repeat. This is kind of a summary, which is the differences between temporal replicates are a combination of the real temporal change, which is the thing we actually care about, which is why we're studying range shifts, and differences in methodologies, which is nuisance. Now, what it means to study this is that we need intratemporal replicates to distinguish between the two. If we do not have intratemporal replicates, if we do not have repeat surveys within a time period when we assume that the state is static, then we cannot distinguish between this bias from methodologies and what we're trying to study, of which is actual range shifts. So as much as we can find data and create these replicates, intratemporal replicates, we're strong. So with that, we can actually take this and we can say, okay, we want to understand font size. If we have intertemporal replicates, we can now separate out the differences in methodologies and biases over time to look at how font size changes. So with that, I will finish, and I think I have maybe one question. So thank you.